Operon Project. A gene can be defined as a DNA sequence that specifies one or more sequence-related mRNAs or polypeptide chains. In other words, a gene can be expressed to produce a final functional product that may later yield a direct phenotypic effect. A regulator gene encodes for proteins that regulates transcription initiation of DNA. It is expressed continuously, although at a low rate, so that a few regulator proteins are always present. Regulator proteins are important for metabolic coordination and gene regulation, so that the proper proteins and RNA are synthesized when needed in the right amounts. RNA polymerase synthesizes RNA. It binds to a transcription start point in a precise location and orientation on the promoter, or a DNA sequence that initiates transcription. After attaching to transcription factors, it moves along the DNA, continuing to untwist the double helix and adding nucleotides on the 3' end of the growing RNA molecule. Ribosomes facilitate specific coupling of tRNA anticodons and mRNA codons. They are made up of a large and small unit of proteins and ribosomal RNA. Once assembled, the ribosome is in charge of synthesizing the polypeptide based on the mRNA sequence. An operator is an on-off switch for a certain group of genes. It is usually located within the promoter, but can be found between the promoter and the enzyme encoding genes. It controls the access of RNA polymerase to the genes. Structural genes are genes that code for the protein and are controlled by the operator. Together, the operon is the entire stretch of DNA required for enzyme production. It consists of the operator, the promoter, and the genes that they control. The amino acid tryptophan is synthesized from a precursor molecule through enzymes encoded by TRP genes. When the bacteria lacks this particular enzyme, it is able to synthesize it through this pathway. Genes encoding for all the enzymes in this pathway are grouped together with a single promoter. These genes are controlled by an operator in the TRP operon. The TRP operon is switched off by the TRP repressor, coded for by the regulatory gene TRPR. Each repressor protein is specific to its operator. The TRP operon is usually activated, since the TRP repressor is an allosteric protein synthesized in a conformation with little binding affinity to the operator. This means that enzymes that synthesize tryptophan are able to be formed. In presence of tryptophan, the TRP regulatory protein is able to bind to tryptophan, changing its shape so that it is able to bond with the operator. This prevents the RNA polymerase from synthesizing the RNA needed to make the TRP enzymes. Tryptophan functions as a co-repressor, a small molecule that functions with a repressor protein to switch an operator off. The binding of a repressor to the operator is reversible. When there is no tryptophan present, then the repressor is no longer able to bind to the operator, and the synthesis of these enzymes are able to continue so that more tryptophan can be produced. The TRP operon is said to be a repressible operon because the operon is usually activated until repressed by a repressor protein. These enzymes are known as repressible enzymes and generally function in anabolic pathways. A mutation of a regulatory gene can result in the repressor not being produced correctly, and the TRP operon is unable to become inactivated. This could result in an overproduction of tryptophan. On the contrary, a loss of a promoter on the operon could prevent tryptophan from being synthesized at all. This is because RNA polymerase is unable to transcribe the mRNA needed to code for the enzymes needed in tryptophan synthesis. An enzyme needed to break down disaccharide lactose, or milk sugar, is beta-galactosidase, and is synthesized with the LAC operon. A regulatory gene codes for LAC-I, an allosteric repressor of the LAC operon. The LAC-I is active by itself and is able to bind to the operator, switching the operon off. This means that the operon is usually inhibited. When inactivated, it prevents the transcription of the genes needed to make the enzymes used for hydrolyzing disaccharide lactose. In order to turn the operon on, an inducer needs to inactivate the repressor. In the LAC operon, this inducer is allolactase, an isomer of lactase formed in small amounts from lactose that enters the cell. This acts as a signal that there is lactose present. Allolactose binds to the LAC receptor and alters its conformation, nullifying the repressor's ability to attach to the operator. This allows mRNA transcription for lactose-utilizing enzymes to proceed. The LAC operon is an inducible operon because it is usually off but can be induced when a specific small molecule interacts with a regulatory protein. Hence, these enzymes are known as inducible enzymes and are generally used in catabolic pathways. A mutation in the regulator gene would prevent the repressor protein from being produced. Therefore, the lac operon will constantly be activated. A loss of a promoter on the lac operon would result in the beta-galactosidase enzyme being unable to be synthesized, so the bacteria would not be able to utilize disaccharide lactose. The regulation of both TRP and lac operons involves the negative control of genes because the operons are switched off by the active form of the repressor protein. In the TRP operon, the operon is switched off in the presence of tryptophan, which prevents the synthesis of more tryptophan. However, in the absence of tryptophan, the TRP operon is able to produce more tryptophan. In the lac operon, the operon is switched off when there is no disaccharide lactose, preventing the production of an enzyme needed for the bacteria to utilize the lactose. However, in the presence of disaccharide lactose, the gene is switched on and is able to produce the enzyme to hydrolyze the lactose. Thank you for listening.